minutes of airtime, in which somewhere around 5 minutes for the panelists to present their material, and then about 6 or 7 minutes for Q&A to maximize engagement with all of you, the attendees. And the presenters, the panelists, should go ahead and share your screen. I would imagine you have some visual aids or some slide deck to share with the presenters. So with that, I'm going to turn it over to Dr. Catriona Jamieson, who's going to be, who represents UC San Diego Department of Medicine. She's a professor of medicine in the Hem/Onc division, and she's got a number of other roles here in San Diego. She specializes in myeloproliferative disorders and leukemia, and her goal is to find more selective, less toxic therapies for these myeloproliferative disorders. Today, she's going to be talking about a splicing modulator targeting cancer stem cells in acute in AML. So with that, what I'd like to do is turn it over to Dr. Jamieson, and I'm going to also share in the chat function a little bit more about her project, and I'll give you the URL for the serum. So with that, Dr. Jamieson, go ahead and over the next 5 minutes and tell us what you're doing.
Yeah, thanks so much, David, and to everyone here. I think we've got a rather an august crew to go through this concept and really talk about what I call the Pluto La Vie strategy. So our daughter is 20 and very sophisticated, and she read this poem by André de Fusco, which is all about live life to the fullest. And this is what I notice our patients can't do when I'm in clinic, because they feel like they're not going to be able to live a full normal life because their disease, a pre-cancer stem cell driven disease, may progress. And those are the myeloproliferative disorders or neoplasms that really derive from a stem cell, but then acquire the capacity to survive and self-renew. So in this space, we've managed to make quite a lot of compounds. As you know, I was a co-founder of a company called Impact Biomedicines together with John Hood, Raghu Sarapelli. We made a JAK2 inhibitor that had really originally been developed locally by Target, and we basically merged with Celgene back in 2018. This is now FDA approved as one of the 2 standard of care JAK2 inhibitors for this disorder called myelofibrosis. So we're seeing very nice responses to this now FDA and European Medicines Agencies approved drug called Inrebic. But for some people, they progress and it's just not enough for them, and they go on to develop full-blown cancer stem cells. So we've looked at imetelstat, which is a telomerase inhibitor, CD47 inhibition. I was involved with the original discovery of that don't eat me signal being overexpressed in these myeloproliferative neoplasms as they progress to AML. And then ultimately worked with Pfizer locally to develop a drug called Glasdegib, now FDA approved as Durismo, which extends lifespan for people who are older or have comorbidities with AML. So really we started saying, why are people getting these mutations in the first place? Why are some of us predisposed to do this? And we found that there are these primate-specific enzymes called APOBEX that seem to be activated during progression of myeloproliferative neoplasms. There really has been brilliant work done on this by Ludmila Alexandrov, Mike Stratton, and others. And really what we've done is, Ludmila Alexandrov is local. We started to look at, is this antiviral mutator that is activated by inflammatory cytokines actually activated in these MPNs at the stem cell level? And the answer is yes. And it's one very specific one, APOBEC3C, and upregulation of this enzyme leads to activation of another one called ADAR1. And together they work to promote leukemia stem cell generation and ADAR activation. So we know that adenosine deaminase RNA1, a lot of you on the call are interested in this as a therapeutic target in terms of the RNA therapeutic space, because it edits RNA and enhances the cloning capacity of malignant stem cells. So we can dial down ADAR transcription with that JAK2 inhibitor I mentioned, fedratinib, also with dasatinib in BCR-ABL positive cells. But really we need to understand, when is this active and can we intercede before it's activated? We've done whole genome sequencing, whole transcriptome sequencing, found patterns of APOBEC-induced mutagenesis. But the bottom line of this talk is ADAR and APOBEC co-localized. They work together during the malignant transformation step. So we want to target both ADAR and APOBEC. We know they activate key cell cycle regulators like CDK13. And more importantly, as you see in the string interactome, can activate STAT3 splicing. So as David alluded to, we've really worked on splicing modulation because one of the effects of this RNA editing enzyme is it can perform intronic editing that leads to splicing. So what we found in the transition from pre-leukemia stem cells to leukemia stem cells was upregulation of a STAT3 beta isoform in the leukemia stem cells from patients with the highest engraftment potential in immunocompromised mice. We can dial down ADAR with these two JAK2 inhibitors because JAK2 STAT3 leads to ADAR P150 transcription, and we can also decrease phospho-STAT3. You see ADAR P150 correlates with that STAT3 beta, but we can't turn it off completely. And I think when we get to the leukemia stem cell phase, we're going to have to turn it off completely. So this is the bottom line of the paper we just published a couple of months ago. For pre-leukemia stem cells, they get started because of inflammatory cytokine activation of APOBECs. Why hasn't this been discovered before? Because you have to do whole genome sequencing on stem cells to see this, and it has to be human stem cells. In terms of the transition to leukemia stem cells, we see an increase in ADAR P150. Now, I mentioned that it changes splicing, and this is the key. We can intercede with this disrupted splicing with a splicing modulator called 17S-FT895. This is being funded by the California Institute for Regenerative Medicine, the NIH, more recently NASA, if you can believe it. They're very interested in turning back the clock on this pre-malignant stem cell formation. 17S is a platinolide-derived small molecule that my colleague Mike Burkhardt in chemistry here made originally, and then we had a serum grant together to do the pre-IND enabling work. It binds at the very core of the spliceosome, which again is disrupted in leukemia stem cells and represents an inherent therapeutic vulnerability. And the way we measure on target modulation is we look for the GFP to RFP switch when we add this molecule to leukemia stem cells. And basically that shows that we're getting on target intron retention. We see editing and splicing dysregulation in other diseases, again, ADAR driven. This is multiple myeloma. A third of patients have overexpression of ADAR1 through amplification at the locus and activation through IL-6. And then finally, there are other small molecules. Antisense oligonucleotide inhibitors are another way to go. So if we wanted to target ADAR, we don't just have to develop a standard small molecule or a PROTAC strategy to enhance degradation. We could actually potentially have an antisense oligonucleotide strategy as we did with Ionis. This is why NASA is interested. When Scott Kelly came back from his almost year-long mission in space, he was 2 inches taller than his brother, but he also had pre-leukemic changes in the bone marrow with translocations, inversions, and activation of inflammatory cytokines and telomerase. So we've developed an integrated space stem cell orbital research lab with NASA funding to be able to study this pre-cancer stem cell development in an accelerated time frame because they have 100 times more radiation exposure in that environment. And so we're developing bioreactors so we can put in our reporters of malignant stem cell activity. So ideally with this splicing modulator, we want to get back to normal hematopoietic stem cell behavior. And so we've got the pre-IND funded again by CERM. The FDA was very interested. We've got composition of matter IP. We've scaled up production. We think it's scalable. We're set to start the IND enabling studies. We have method of use IP and have started a company called Aspera Biomedicines. And that company is co-founded by our Impact co-founders, as well as Mike Burkhardt and Sheldon Morris, to further develop the chemistry and then also the clinical trials, where we anticipate this is going to be useful for the 28% of all cancers that have disrupted splicing through MYC activation, and a lot of cancers that have mutations in splicing factors like SF3B1 and U2AF1. So that's it for me. I'm happy to take any questions. We have a uniquely collaborative environment here, so I'm happy to take questions.
Thank you, Dr. Jamieson. Once again, if there's anyone who has a question, please go ahead and put it in the chat. We'll go ahead and we'll read that. But just one question for you, Dr. Jamieson. So you had mentioned It's Aspera Biomedicine, which
Yeah,
is taking this forward.
ASPERA, Aspera Biomedicines. And
Perfect.
that's from Paraspera ad Astra, through hardship to the stars. So everybody knows on this call, it can be sheer hardship to try and make a new small molecule biologic or therapeutic strategy to block
Right.
cancer cells.
Yeah, can you just comment on the, just overall on the timelines for the pre-IND, and then when do you anticipate to make an IND submission
So
for
the
this
pre-IND,
approach?
yeah, we've done the pre-IND already, so the FDA liked it. It was targeting acute myeloid leukemia that had evolved from MPNs or MDS. So they like it. They actually suggested that it's such a potent compound, they would want a modified Fibonacci schedule, meaning you can't more than double the dose between cohorts. So we've written the clinical trial for that. I think it'll take us 18 months to get to clinic. We've already set up with all the CROs to start the process of the IND enabling studies. So we've kind of got that good to go. And then, you know, for the phase 1, we'll do a phase 1 safety tolerability proof of concept study with the splicing as our readout. So we have splice isoform biomarkers of response. So we'll employ those in the clinical trial and then be able to expand after 6 months to other entities, provided we see a good safety signal.
All right, perfect, perfect. Once again, if you have any questions, go ahead and I do have a question coming in for Dr. Jamieson. Can you use downstream splice variants as biomarkers to prospectively select patients in trials?
Yeah, it's a great question. A lot of the trials done with previous splicing modulators like H3B, 8800 or E7107 use splicing factor mutations. But what we're finding is that there are splicing alterations in the absence of splicing factor mutations, and that's in the setting of ADAR activation and other mechanisms. So yes, we can use MCL1 long, SF3B1, SF3B3 isoforms, P2K2B, so CD44, 012. These are leukemia stem cell overexpressed isoforms that we can track. The other thing I should say for the FDA, they were quite keen for us to work on pediatric indications. So we're also seeing acute myeloid leukemia in kids as a potential major target for this splicing modulator. So yes, we can stratify and do a molecular stratification by the splice isoforms, and we'll build that into the clinical trial design. Great point.
Great. Thank you once again, Dr. Jamieson. And if you have questions, feel free to reach out here or directly to Catriona. That would be wonderful.
Thank you.
Our next presenter, I want to make sure that we get everybody in on time here. Dr. Theodore Nowicki, who's MD, PhD from New York Medical College, did his residency in pediatrics at UCLA School of Medicine. He is affiliated with the Hem-Onc group in the pediatric division of Hem-Onc. And we also have an award winner here. He's in 2021, he was named a super doctor, Southern California Rising Star. So congrats on that. I'm going to go ahead and share, let's see, Dr. Nowicki's research also on the chat through serum, but I'd like to turn it over to Dr. Theodore Nowicki. Tell us a little bit more about his work.
Well, thank you so much for that introduction. That was very kind. So I'd like to just give everyone a real, solid 30,000 foot view of cell therapy pitfalls and challenges, and how we're trying to overcome them with a stem cell based therapeutic. So sort of a different take on stem cells than we just heard here. We're trying to actually use them to our benefit. And so generally, when you're talking about cell therapy paradigms, we talk about them a lot. You've seen great successes with the CAR T cell platforms for the treatment of liquid malignancies, which we call leukemias and lymphomas and so forth. But treating solid tumors with cell therapies is a little bit of a different paradigm than you see with liquid tumors. And in general, just for those that might not be familiar, the way that we generate these products is you have retroviral or lentiviral mediated transduction of a patient's T cells to express a tumor antigen specific T cell receptor, or in some cases a chimeric antigen receptor like a CAR, which will then seek and destroy preferentially a tumor cell population that expresses the antigen of interest. And in general, these are made from a patient's own T cells. At UCLA, we do it in-house kind of in real time while they're being conditioned. And the important thing to really take away that you don't really need to be a radiologist to appreciate is that these patients have really robust, really substantial responses to these therapies within the initial wave of activity within the first couple of months or so. Here we have on the top a patient with melanoma who received the F5 MART1 transgenic T cell receptor, and seeing some dramatic regressions of all of the lesions here on PET. And then here we have the NY-ESO-1 TCR for the treatment of advanced sarcomas, which are my specialty, which are soft tissue and bone tumors. And you can see the metabolic activity here representing the tumor life is basically melting away within the first couple of months here. And unfortunately, the main problem with these therapies is that you have this great honeymoon period where the tumors are regressing really substantially, but they're not really able to have a very robust long-term response rate, unfortunately. So you have a significant proportion of patients who don't respond at all. These are the ones shaded in white. These are the patients with melanoma that we treated on our trial here. And here representing the time of disease progression, and with the X here with time on the Y-axis in months, or on the X-axis in months rather. And then the remaining patients shaded in red here did have a transient response here coded with the diamond, but unfortunately within about 6 to 12 months, generally these responses are not sustainable and the tumors ultimately stop shrinking, stabilize, and then start regrowing, unfortunately. And with sarcomas, the other type of tumor that we study here, same song, second verse. You see a subset of patients that don't respond at all, and then you most distressingly have patients who respond, but in a very transient manner, even though it's very dramatic at first. And so the work that we generally focus on in my group is what are the pitfalls? How can we make them better? And in terms of the pitfalls, one of the things that we've noticed over time is that these transgenic T cells don't persist very long term in circulation. Here on the y-axis here, you have the proportion of T cells in circulating that possess the transgenic tumor-seeking TCR, and then time here on the x-axis. And you can see that in general, you have this initial spike within the first couple of months, but over time, the persistence of these T cells, unfortunately, is not really sustainable. And that tends to also recapitulate in the patients we treat with sarcoma as well. And these drop-offs in persistence is generally correlated with the time at which these T cells start to, at the time at which these tumors start to regrow. But beyond just persistence, the other thing to remember is that age of the T cells matters. Within our body, we have T cells running the gamut of phenotype. I hear here from youngest to oldest, naive to memory to effector, exhausted. And basically, as T cells age over time, they become less effective at fighting infections, or in our case, fighting cancers. So in general, you really like to have this sweet spot over time. And so unlike a conventional molecule where you give it and it metabolizes and then it's passed, these are living drugs. These are cells that unfortunately, have a finite lifespan, even if they are persisting. And because I'm pediatrics, we like to do cutesy analogies. So I like to say that the naive T cells are like the young Steve Rogers with a lot of fire in his belly, wants to fight the Nazis. When you get to effector, that's when he's in his prime. That's Captain America, able to, really do some damage. And really, by the time you get to effector exhausted, he's, wiser, certainly, but, not as physically robust as he once was. And that's very classically associated with the T cells as well. So I've told you all this, but the question is, how do we overcome that? How do we get, surmount these? And we've tried to move the needle in a couple of different ways, but the one that I'd like to talk about today, which is funded by CERM, is transducing the TCR both at the level of the circulating T cells and at the level of the hematopoietic stem cells as a means to provide a long-term renewable source of transgenic T cells, which will not only be long-term persistent, but will also be a long-term source of younger T cells as well. So this is sort of our oversimplified version of how this works. You have this initial honeymoon period, like I showed you in the previous patients, where you have this initial great wave of activity, but unfortunately it doesn't persist. By the time it stopped persist at several months, that's when you're going to have the progeny of the transgenic TCR T cells that arise from the bone marrow hematopoietic stem cell niche actually entering into circulation and actually causing a sustained degree of clinical response. And in terms of how this works, as I mentioned before, we basically are co-administering these. So we have the traditional retroviral modified T cell product, which just encodes the TCR of interest, in this case against MYSO1. And in parallel, we're also transducing the patient's mobilized peripheral blood stem cells, which are mobilized from the bone marrow, to include both the transgenic TCR as well as a SO39TK, which is to both image the gene progeny and also serves as a suicide gene functionality as an added safety feature as well. We can ablate these cells at will with ganciclovir if we so choose. And the overall way that this works is that this is a very oversimplified cartoon, but just to make sure that everyone sort of has understanding the 30,000 foot view here, we transduce the T cells, the TCR at the level of the blood stem cells, and it percolates all the way down through every lineage that arises from the bone marrow. But because it's a T cell receptor, it's only able to be functionally expressed, trafficked to the surface, and actually have activity within the T cell lineage as well. Everything else, the DNA is there, but it doesn't really do anything. It's largely vestigial. And just to show you one of our clinical patients that recently received this was a patient with metastatic synovial sarcoma that had progressed after multiple lines of therapy. I won't really get into the weeds here, but these are some images of her lung lesions here, which you can see, dramatically shrinking in that honeymoon period I mentioned before over time from baseline to day plus 53 here. But here is the day 25 FHBG PET, which is a specialized tracer which can visualize the SO39TK product of the T cells. And here you can see that this is showing engraftment within the bone marrow niche of the patient to provide a long-term renewable source of these here. And so I don't want to take up too much time. I want to leave time for questions, but I'd like to acknowledge my group, Tony Rivas, who really gave me a chance when I was just starting out, and everyone else in the group that helped develop the tech, specifically Owen Whitty, Don Cohn, Jerome Zach, and Lily Yang, as well as everyone else at Caltech, David Baltimore and Jim Heath, late of Caltech, and also our great clinical recruitment team, and also the rest of the clinical team and pathology team that we work with. And I'd love to also thank our funding sources as well, both to me and to my group, and I'd love to open it up for any questions that anyone has.
Dr. Nowicki, thank you very much. And I love the pace of this. Dr. Jamieson, Dr. Nowicki, we all have our seatbelts on, just, going through these. These are really wonderful. I had a question, and once again, I encourage everybody, if you'd like to ask a question, go ahead. You had mentioned, so the two pitfalls: persistence, the T cells are dropping off, and then the age of the T cells. And then the way you're overcoming this is you're transducing the TCR on the hematopoietic stem cell level. Is there anyone doing this clinically currently today, or I should say commercially, that we're looking to either replace or do differently? How are others, approaching this issue?
So in general, this is the only phase 1 of its kind in the world today. So, and the big reason for that is because I was trying to give a sort of 30,000 foot view of a lot of this, but there are, a lot of clinical challenges of treating a patient with a stem cell based treatment as opposed to a traditional T cell treatment. There are, issues with that. And again, this is phase 1. So, a lot of these we're learning kind of as we go. Right. Because when you're taking a patient and giving them basically an autologous bone marrow transplant, which is what we're doing here, that's a very, very different clinical situation than a patient who is just getting a cell therapy, which basically involves completely different types of conditioning chemotherapy. You're ablating the bone marrow. These are patients that have to stay in the hospital for, I usually tell patients, and I consent them, like, plan for a month. If we do better, then that's a bonus. But yeah, so we are the only ones in the world currently doing this exact type of work.
Great. And John D., you have a question?
Oh, you're muted. Sorry.
You're in mute, John.
We still can't hear you. Sorry.
How about now?
Yes, that's perfect. Thank you, John.
Great. All right. Yeah. Thanks, Theodore. Curious, what's the kinetics like going from the stem cell state into, a T cell that can actually do something?
So, I mean, as I sort of showed you the simplified version earlier, but that is actually pretty classical, because when you have T cells arising from the progeny, or T cell progeny arising from the bone marrow, that does actually take a few months to really, ramp up to speed to the point where you're seeing functional product. And part of that has to do with just the kinetics of bone marrow recovery whenever you give a patient a bone marrow transplant. In this case, it's an autologous bone marrow transplant, meaning it's their own cells, so that does help as opposed to giving a donor. But the other thing is that when you give a lot of chemotherapy, you wind up really damaging the thymus as well. And that's, if you recall, T cells need to sort of be tolerized through the thymus. And so you need to kind of have both pieces in place. So it's the bone marrow, but we're also finding that it's also the thymus that needs to really recover as well. And, collectively, those processes do take, at least 3 months at a minimum, and probably slightly more.
So that explains why you're doing the co-admin of the T cells as well as the stem cells. Okay.
Exactly. If we just gave the stem cells in a vacuum, it would be like we've just wasted 6 months at least, because the patients
they could progress in the meantime.
Well, and they already need to have a washout and everything for this. Yeah. So you really want to give them some kind of honeymoon period like we've traditionally given.
Great, thanks.
Great, good questions, John. And I just had one other question. What's the status and the nature of the IP?
So, the IND is, locked and loaded. We did all the pre-IND work about 5-6 years ago, I want to say. Like I said, we're phase 1. We've dosed, several patients currently. The big thing we're doing right now is we're actually in the process of modifying the vector to omit the suicide and imaging gene, the SO39TK component, to improve transduction efficiency, and also, again, without getting too detailed, but we found that it winds up really, when you're talking about prophylaxing against viral infections, not having the ability to give acyclovir or ganciclovir as prophylaxis for viral infections really does complicate things, because the only thing, these are pills typically, and so if you have someone with a reactivation of say, like zoster, let's say, with a bone marrow transplant, classically, you can just give them acyclovir and call it a day to prophylax against it. Well, here, you can't really prophylax anymore, so you have to sort of wait for it to happen. And if you do have it happen, then the only options are to give IV medications. There's different classes, but basically you've automatically talked yourself into getting an SAE because now they're in the hospital. So, so that's another reason why we're kind of like, okay, we've decided it's safe enough to remove it. And the FDA actually had wanted us to remove it by the end of 6 patients anyway. So we're just doing it a little early right now. And yeah, so we're just going to get that signed off by them, and we're still continuing.
What about the intellectual property? Where are we there?
So it's not something that I personally own. I'm the IND holder, but basically all of that is through UCLA, since that's where the tech was developed. So if anyone wants to talk about that and sort of the nuts and bolts of that, I can put you in touch with our tech development people.
Very good, very good. Well, I didn't see any other questions coming through. So with that, I would like to thank you, Dr. Nowicki. John, your hand is still up. You still have another question?
Oh, that's funny. I did have another question, but I was going to politely.
I felt it, John.
Yeah.
Last question.
Theodore,
last question.
I'm curious if there are other approaches. There's been so much work on like TCF7 and T cell stem-like states and epigenetic modeling. Are there other sort of small molecule approaches to try to preserve that stemness?
So that's a great question, and that's actually a completely parallel body of work that I do, that I have other grants to fund in my lab. Is basically my big squeeze, in addition to all this, is focusing on epigenetics and the epigenetic, because those are ultimately what we find to be the most salient changes that trigger T cell aging. And if you can, tweak the epigenetics or pump the brakes on them a little bit, you can extend the lifespan of these cells. And the short answer is that we found, we recently published in Cancer Discovery some work showing that there are certain DNA methylation signatures that actually can impact the persistence of the vector in just T cell products, not talking about stem cell products. And so the work we're doing right now is trying to actually look at small molecule inhibitors, DNA methyltransferase inhibitors, to see if those can possibly extend the lifespan as well. And that's also attractive because some tumor markers like NYSO1 actually can have their expression upregulated when you treat them with things like decitabine and stuff like that. The problem, unfortunately, is that you run into a lot of toxicity, and you also wind up not really having an elegant answer when your IRB asks you, well, what's the timeline? How long are you going to give these people decitabine? Like forever? And the answer winds up being kind of forever for now. So the short answer is that, yes, we think that's also probably an option. It's probably ultimately a little bit more complicated where the rubber hits the road, however.
Okay, thanks.
Okay. All right, you got it, John? Good job. Dr. Nowicki, thank you very much for your time.
Thank you.
All right, with that, we're going to move over to our third presenter, Dr. Tsui-Fen Chou, who's a research professor of biology and biological engineering at Caltech. She's going to be talking about small molecule inhibitors of AAA ATPase proteins. And with that, I'm going to turn it over to Dr. Chou, and I will also put into the chat a little bit more about her research, the research going on in her lab. Dr. Chou.
I cannot share. The other one need to unshare.
I'm sorry, you're trying to share?
Yeah.
Sorry, Dr. Nowicki has to stop sharing his screen.
Okay, Dr. Nowicki. Yeah, okay.
Sorry, I was trying
You
to.
can give my talk if you want.
It's very much a control thing.
Sorry about that.
No problem.
Go
Hi.
ahead.
So can you at least see the screen, the slide
Yes,
now?
we can. Go ahead.
Okay, perfect. Okay, thank you very much for this opportunity. So just a little bit background, like my training is in Taiwan. I did my pharmacy and I moved to University of Minnesota to do my PhD in medicinal chemistry. Then I moved to Ray's lab at Caltech to do my postdoc. And after all of this training, I started my own lab at Harvard UCLA for like 7 years. Then I moved to Caltech 2019 as a research professor. So since I only have 3 to 5 minutes, I want to make sure I thank everybody who actually brave to join us 7 or 8 years ago as a young faculty. And those are the people actually make all of my research possible, and also my collaborators and the funding from NCI or NINDS, and NICHD, and NIA. And recently, our work for P97 in antiviral research is funded by Merck Institute for Translational Research at Caltech. So I will only focus on 1 or 2 main projects that we target 2 proteins. And those proteins are called AAA ATPase. The AAA actually stands for ATPase associated with diverse cellular activity. So basically it means they're doing multiple things in the cell. And the one on the left is called P97. In yeast called CDC48. In gene name is called VCP. The one on the right is called RAP. And there are also 2 alternative names. And you can see, structurally they look very similar. They form a hexamer and they have different domains. And in the cell, how they perform different function actually is through interacting with different proteins. So you can imagine inside the cell, there are 10 to 20, 30, we still try to study different flavor of those proteins. It depends on what it's supposed to do, depends on the signal, depends on the stress. It will find the right interacting protein. And those interacting proteins move them to the correct location to perform its function. So this is the summary of the project in my lab currently. And the first 3 highlights, that's what I will describe a little bit. And you can see basically, for the cancer, P97 and the RAP are potential target. And we already have, for P97, it's developed longer since I was a postdoc, so there's more story. For RAP, we basically have some early compound, and we have provisional patent to protect that, and we also try to get NIH grant to support more work. And the second project, like I mentioned, is funded by Merck Translational Institute that we show by genetic and small molecule targeting P97 can be effective for coronavirus. And those are still in the cell stage. And we would like to be able to get funding to do animal study or hopefully be useful for future outbreak. The third one is a small molecule inhibitor or activator. And this is still related to P97 because P97 mutation, the single nucleotide change causes single amino acid change. And this causes a rare disease called IBM PFD or ALS. So this, we have our one to perform high throughput screening in collaboration with NIH, NCAT. So we have some early compound and we would like to do more SAR to really show it's effective to animal. And of course, hopefully to clinic. So the fourth project is relevant to the third one. This is also funded by R1. There we use patient-derived iPS model to test the compound we found, and also to do proteomics to figure out what are the potential disease biomarker we can use. And the fifth one actually was more exciting these days because I'm now a faculty director for a facility called for mass spec. And we believe we need to develop a good method to look for protein sequence variant in almost all of the possible disease or sample, cancer, neurodegenerative, even virus, because virus can modify RNA. They cause the protein sequence, and there is no way to detect that at genetic level. And the mass spec, the software currently available, we cannot detect. So we really need to develop a good method. And basically, it's a computational limitation, but we have an idea to try to avoid that by have a pre-compute database. And hopefully, once we develop that method, there will be, we can advance the mass spec research and also for disease implication. The sixth one is new, like we want to develop like PROTEC, but we want to target the protein to autophagy for degradation. The last one is enzyme replacement therapy for neurodegenerative disease. So the approach we use, I kind of scan through it. We do structure-based design, virtual screening, high throughput screening, chemical proteomics, affinity mass spec, also thermal proteome profiling to find on target and off target. Also, we try to isolate resistant mutant for the compound we find to see if it's really on target when it causes cancer cell to die, and CRISPR and also iPS model. So here is the project that I started when I was a postdoc, and we start to target P97 as oncology indication. And you can see I put a timeline here, just since 2001, and before that I already spent like 2 to 3 years to find these hit compounds through high throughput screening, and we have different collaborators help along the way. And Ray started a company called Therapeutics, and these are the current status of the 2 compounds. One that enter phase 1 clinical trial, one failed because of target fail. Luckily, there's another compound that we can show there's no off target effect. So now the active compound in phase 1 clinical trial is called CB5339, and now it's in AML for Cliff sponsor, and also all comer in NCI just to see what are the response. And in my lab, we try to characterize the resistant mutant arise from these compounds and try to identify ways to tackle those mutant cell, because all of the target therapy, the relapse comes, then how do we help the patient? And we use the approach I just described previously and try to figure out what can we do. And recently we just published this paper. We try to target the cysteine active site on P97. So you can see this is a covalent inhibitor, and you can say, there might be other potential off target. All of these will come, but we need to find the right molecule and have the right study to show the compound is not targeting every other possible cysteine on the protein, right? But no, these are the first step to achieve that. Mm-hmm. Then the important thing is, P97 has its normal function. So this is an idea really we want to realize, is to find a specific P97 complex that is always expressed in certain cancer. So when we target that particular complex, we will not affect the normal function of P97. And this is ongoing, a lot of proteomics work, use different approach, see how do we reach the end chemistry. We have so many different compounds in hands. Once we have the right assay, then we can test them. So the small molecule activator, this is a high throughput screening is done. So we want to find selective compound for the mutant. So we don't want to hit the wild type in the cell, wild type P97 in the cell. And summary is there's a complex we believe is more disease relevant. That's why we select this complex. And this is the IPS model. Since those, we know for neurodegenerative disease, good model is important. And we already make those model and we already test the small molecule Cliff developed can actually, at lower dose, you don't want to give too high a dose because then you'll kill the cell. At lower dose actually can protect the motor neuron from death. So those neurodegenerative disease is the neuronal cell will die, but how do we help them to survive longer? And hopefully this can also be useful in the future. And these are just the model why we believe we want to spend time essentially to develop this and to do more biomarker and disease treatment screen. And that's it.
Thank you, Dr. Chou. You mentioned ALS. Are there other neurodegenerative disorders that you're going to target? Alzheimer's,
Yes,
Parkinson's?
Yeah, I didn't get any background for that. I need 3 minutes. So,
I
so
understand.
basically, P97 mutation cause ALS-like, but also they are frontotemporal dementia. And so one of the grants actually is found through the ADRD, Alzheimer-related dementia. So it just depends on the patient. And that's why the IPS is not focused on one cell type because 90% of patients has the motor neuron disorder, 30% of the frontotemporal dementia. Some of them have the bone defect. So it's a complicated disease, and we believe through the multiple function of those AAA ATPase, and we want to be able to link which one to which marker from mass spec. Yeah.
Very good. Anyone have any questions? I have one more, but I just want to make sure I'm respectful of other people's questions. I'm not seeing anything. Are there other P97 inhibitors either in the clinic being developed or on the market?
Right. So the one from Click Bioscience, that's the one only the two enter clinical trial. So there's one was developed by Genentech, but I think after they publish, at least from what I understand, my understanding is that they don't have any development yet for clinic. So they stopped in the stage, I think 2013 or 2014. Yeah.
Great. Well, I appreciate it. I'm not seeing any other questions coming in. I appreciate all the great work you're doing. A lot of chemistry going on there. So thank you very much. And it looks like we're just about on time, which is wonderful. At this time, I wanted to turn it over to Dr. Yang Zhang. Dr. Yang Zhang is coming to us from the USC School of Pharmacy. He's going to be talking about the novel immunotherapeutics using synthetic exosomes. I will go ahead and share a little bit more about his background and his research in the chat function. And without further ado, let's turn it over to Tiger Zhang.
Okay, thank you, Dave, for the nice introduction, and thank you for the organizer for giving me this great opportunity to briefly present a very exciting technology that has been developed in our lab at USC. So my lab is very interesting to learn about this cell-derived nanocarrier named exosome, also known as the extracellular vesicle. So the exosome can be derived from virtually all the type of different cells. Through these two round of these memory evaginations, they can form at the end of internal of this multivesicular endosome, followed by the secretion into the extracellular space. So these cell-derived nanocarriers, they contain a variety of different cargo derived from the parental cells, such as the nucleic acid, enzyme, the membrane proteins. So in the past 10 years, exosome has been emerging as a very important topic in the biomedical research. More than 10 years ago, people believe that exosome just kind of like recycler for the cell garbage, they do not play a very important role. But in the last 10 years, there's a growing increased attraction for this exosome research, because for the biological part, the exosome has been shown they have very important role to mediate the cell-to-cell communication through this transportation of the cell cargoes as well as the direct fusion with the target cells. And in addition, given their potential to deliver the therapeutics, exosomes have already been used to deliver the different type of therapeutic. Just in last year alone, there are more than 4,000 papers published focused on the exosomes. So compared with other different nanocarriers, the exosome has some very unique properties. So unlike the synthetic nanoparticles, the exosome, they are enriched with the membrane protein-derived protective cells, such as the CD9. This type of tetraspanning, they can facilitate the direct fusion with the target cells, which can enhance the delivery of the cargoes. Unlike the synthetic nanoparticles, those carriers usually be trapped in the endosomes and need some other ways to escape for the therapeutic deliveries. Moreover, on the exosome surface, there are some like CD47 derived from the parental cells. This CD47 could send the don't eat me signals that can prevent the clearance by the monocyte as well as the macrophage, which make the exosome be more stable compared to the other nanocarriers. If you compare the exosome with a virus, which can induce like the immune response. In comparison, this mammalian or the human cell-derived exosome, they have a strong potential to minimize the immunogenicity. So given their high potential for the drug deliveries as well as the extended stability, low immunogenicity, so exosome has a very high potential for the therapeutic development. So many studies has been done to use the native exosome to deliver like the siRNAs, microRNA, or some other small molecules. In our lab, we are very interested to transform the native exosome to the immunotherapies. So in our case, we play a lot of genetic engineering on the exosome surface to display the two different type of monoclonal antibody on the exosome. So we named this type of like engineered exosome as the smart exosome because they are featured with a multivalently displayed monoclonal antibodies. So these antibodies can specifically recognize the tumor cell surface antigens as well as immune cell surface markers. Through this exosome-mediated cell-cell engagement, that can potentially induce the anti-cancer immunity in a very specific and controlled manner. And in addition, unlike the molecular therapeutic, like a bispecific antibody, this exosome, they still can function as the nanocarriers. They can also be loaded with a payload, such as a small molecule immunomodulators that can be further facilitate these targeted deliveries to enhance the anti-cancer immunities. So just to give you an idea, so we have some proof of concept study by making a smart exosome that targeting the T cell surface CD3 as well as the cancer cell surface EGFR. So to make these smart exosomes, we made the genetic fusion protein that is encoded in these mammalian expression vectors. Following the transfection into the mammalian cells, we can purify this smart exosome. Then we did preclinical studies using the animals with the xenografted human EGFR positive tumors. In our proof of concept study, we can demonstrate this anti-CD3, anti-EGFR smart exosome that are very potent to suppress the anti-tumor immunities. So let's just give you some preliminary data showing that for these smart exosomes, upon injection into this EGFR positive tumors, that can induce the strong anti-tumor immunities with a significant shrinkage of these tumors as well as with not apparent toxicity. So this is the proof of concept study using these animal models. And also we have demonstrated this could be a potential general approach that can be extended to the other different tumor-associated antigen. So in this case, we derived this smart exosome that features the antibody targeting the human HER2, which is a validated target on the breast cancers, as well as the T cell surface CD3. Using this anti-HER2, anti-CD3 smart exosome, we can demonstrate that this exosome would be able to recruit the primary human immune cells to the HER2 positive solid tumor, and in the animals that can induce the tumor suppression. So this is another study. So with that, I want just to summarize the technology we have been developed at USC. So through this genetic engineering, we can simultaneously display more than 2 different monoclonal antibody, which is functional, can engage with a high affinity and specificity to the different antigens. And through this smart exosome that can induce the potent and specific anti-tumor immunities. We think that this could be a possibly general strategy that can be used to target not only for the cancer-associated antigen that can be used to solve other like non-cancer derived indications. So based on all our data, we have already demonstrated preclinical feasibilities by using this smart exosome for the cancer immunotherapies. So this exosome, unlike the previously established in the field, which can become a new type of like nanomedicines. So with this side, I want just to acknowledge my lab member who've been working on this one, as well as our fantastic collaborator, and of course, the funding agencies who has been continuously support our works. And I would like to thank you for your time. I would happy to answer the question you may have.
Edgar, that's a lot of collaborators. Thank you very much. We have just time for one question, and I see John D. Go ahead, John.
Yeah, thanks, Yong. Very intriguing work. I'm curious. It looked like your proof of concept. You had, you know, say a HER2 CD3 or an EGFR CD3 bispecific antibody. Have you established that you can have the 2 monospecific antibodies expressed by the exosome, and whether or not that mediates the same activity?
That's a great question. We do show that by just simultaneously display 2 monospecific antibody, they can also induce some anti-cancer immunity to some degrees, but that one is not as strong as the one where we put this single chain bispecific on the surface.
Okay, thank you.
And Dr. Zhang, Ryan Witt has a question. He put it to the chat. I'll just read it for you. So the exosome is intracellularly delivering an antibody to HER2, or what's the exosome delivering, or the proposed mechanism of action of inter-tumor activity?
And just to follow up, just to follow up and to clarify, is the antibody on the exosome surface, or is it delivering the antibody?
Just to clarify, so the exosome we put on exosome, the antibody was displayed on exosome surface.
Okay, thank you.
Dr. Zhang, just a question, more of a comment. I love the hockey stick growth on the exosome research. It's really, it's almost like logarithmic. Where are we in your research with regards to getting this into the clinic? And what's that first target? Is it breast cancer?
We have some other targets, such as the AML disease, as well as the prostate cancer disease, where we want to extend this to some other, like with some mathematical needs. And in addition, we are trying to further advance this technology by display some other immunomodulatory functional domain on the exosome surface that can be more responsive to this tumor microenvironment to provide more sustainable anti-cancer immunities.
So what I heard you say is that both liquid and solid tumors are both
yes,
amenable targets for.
Correct.
All right, great. John D, last one.
Oh, I'm sorry. I think that's a vestigial hand raise.
Oh no. Well, we're waving back at you. Thank you very much for all your questions. All right, Dr. Zhang, I appreciate you presenting this research. Nice job. And for our last presenter, we're going to shake it up a little. We're going away from the therapeutics now to the diagnostic side, and would like to present or ask Dr. Paul Dempsey, one of the co-founders for Hawkeye Bio, and co-collaborator with André, who I think might still be online. Paul, go ahead.
Hi. Thanks very much, David. André is online. I just driving a car, so I'm able to look at the screen and will hopefully do it justice. Thank you for the introduction and for the invitation. It is a different pace here, but I think there's an overlap in what we're all trying to do, which is to really make an impact on the number of people who suffer from these various diseases. Hawkeye is coming at this from a different angle because we're trying to play a role in detecting cancers earlier. And we're doing that with a nanosensor platform that we're developing down here at the LA Biolab site in Torrance. These are low-cost, distributable, very sensitive platforms that we're focusing initially on lung cancer in order to find a way to distribute an easy test that can help distinguish disease when it can be cured. We're an Irish company formally, although the C Corp is based in California. We also have a presence at the Johnson & Johnson's J-Lab in Shanghai, which will be very important, as you'll see in our story. And the technology is something that we've been working on for a couple of years, but has been patented, and we're in the process of commercializing. That all happens from our site here down at the LA Biolabs, which is a great facility. We're focused on developing the sensors, their targets, and coordinating the clinical studies. Our first and in many ways most obvious target is lung cancer. This remains the largest killer of cancer worldwide. It outperforms, unfortunately, everything else that we could consider, and it's plagued by challenges in detection, non-compliant screening availability, and very different approaches in different regions of the world. So the United States has a US PSTF mandated and sanctioned screening program. That's very different in Europe, and it's very different in Asia. Despite that, if we were able to implement that type of monitoring properly, we could have a big impact on disease. Like most cancers, if you detect it early, you can actually make a big difference in the long-term survival of patients who have cancer. But we do a very poor job of detecting the disease at the point where it is resectable. So we focused on building a low-cost nanoscale nanobiosensor, we call it NBS. These are fluorescent-based sensors that detect protease activity. I hope that my slides are moving forward. Are you all seeing the slides change?
We can see the slides, but at least on my screen, they're not moving forward.
No, well, that's no good.
There.
Okay. So the sensors are nanoscale sensors that detect the presence of activated proteases. They're a carbon-based platform with a specific peptide for each protease. When the active protease is present, it will release a dye that is unquenched when it leaves the carbon-based substrate. So our readout is to measure the increase in fluorescence for each specific sensor. In a lab setting, the test is run by collecting blood from the subjects, extracting the serum, incubating that serum with a panel of protease sensors, each one specific for a different protease. After an hour of incubation, we read out the fluorescence, and that level of fluorescence is integrated to give us an up or down readout for the presence or absence of cancer. Now, our approach, recognizing the fact that different regions of the world have very different approaches to identifying subjects, identifying patients who are at risk and have cancer. We're building up clinical pipelines in both Europe, Asia, and the United States because the clinical path for patients into the standard of care is quite different in each region. At home, those subjects are focused on building the clinical data we need to commercialize this test and the basic platform technology. As I say, we're focused this year and into 2022 on lung cancer. We have cohorts that add up to about 1,800 patients over the course of 2021, looking at lung cancer and focusing largely on the stage 1 and nodules stage of disease where the disease is resectable and can be treated. Meanwhile, here at Torrance, we're manufacturing the sensors and over the course of Q4 and transitioning that production to some commercial partners in Southern California who have GMP capabilities. Just to give a sense of how the approach is playing out in lung cancer, as I mentioned, we're picking panels of proteases that are designed for any particular disease, and these can be deployed in many different cancers as well as inflammatory diseases for the derivation of a lung-specific panel. We have a discovery panel of 19 sensors that we evaluated on a cohort of 350, just over 350 patients that were made up, about 30% of them were from lung cancer with distribution across all stages, and as many of the confounding factors that we could get our hands on for other inflammatory diseases of the lung like COPD and asthma and chronic bronchitis. With our 19 sensors, our multivariate analysis distinguishing all lung cancer from all non-lung cancer, the p-value was less than 0.0001. So we're moving forward with this series of cohorts incrementally over the year to define the subset of the 19 that will go into our first kit. We have processed the next 100 samples. We will be getting the data on that in June, and this will continue throughout the year. From the United States perspective, the USPSTF has a sanctioned low-dose CT screen that is used for people who are over 50 with 20 pack years or have quit smoking within the last 15 years. And this is a tool that's used to find people with lung cancer in the United States. It's a great tool, but it's poorly taken up right now, with fewer than 5% of smokers going through, who meet this criteria, going through the screening stage. And the change this year to decrease the age to 50 from 55 and decrease the smoking requirement from 30 pack years to 20 has greatly increased the number of patients who need to be screened on top of a poor performance already. So we see a very important need to feed that pipeline and to help identify patients and triage them into the screen by deploying this type of technology as a pre-screening tool, where those most at risk will get the most attention to bring them into the standard of care. Outside of the United States, the LDCT is not a standard of care. So most other regions are looking desperately for strong, supported ways of identifying patients who have cancer. When you look at that market just in the United States, the recent change in the screening criteria means that there are over 14 million people who are in the United States currently eligible for LDCT screening. With a generous compliance rate of 8%, that's maybe 1 million people who are being screened, leaving over 13 million who are not being taken care of. So depending on how you price your test, there's a lot of people who need this help, and there's a big market for accelerating the funnel for people into low-dose screening. Outside of the United States, the market's even larger. Europe and China, Japan have smoking rates that are unfortunately much greater than the United States, and that has a commensurate effect on the number of people who require screening. It's our aim to, well, we come back. We've been in China since 2019, excuse me. We won a quick-fire challenge with Johnson & Johnson that gave us the foothold in the JLabs facility that we've maintained. And we're just starting to staff that now as COVID starts to pass us by. But the market in China is emerging and changing rapidly. There are emerging private healthcare models that we've been in contact with who have hundreds of thousands of doctors in their network. With the Tencent Trusted Doctor example is these data are 18 months old now. There's over 30 million subscribers and 40 hospitals that are in that network. And a lot of telehealth and WeChat provided testing is a standard practice So we see that presence in China as a very important part of our focus. Just to put a sense on where we've been, Hawkeye started in 2018. We identified and started working on our technologies in 2019. We closed a seed funding round at the end of 2019 that was led by a global life science company. Over the course of 2020, we developed this nanosensor technology and provided the proof of concept and the initial clinical validation data. And this year, we're focusing on extending our clinical validation to this 1,800 subject pipeline and transferring the manufacture of the nanosensors into professional manufacturing and kitting groups that we're working with in California. We will close a Series A towards the end of this year. And that, I think, is my time. So happy to take any questions.
Paul, thank you very much. And before I was going to throw out some questions, I didn't know if, Andre, I know that you couldn't see the slides, but I'm sure you've given that presentation many times. If you wanted to make any comments online. I'm going to assume no. Okay.
Yes. Hi.
Go ahead, Andre.
Hello.
Go ahead, Andre.
Sorry. Sorry, it took me a second to unmute while I'm driving. I apologize for that. Yes, given the presentation many times, I thank everybody and also Paul for using some slides that were not just scientific, where he's more comfortable describing the principle of the nanobiosensors. You know, we often talk about the underlying technology here, but we're trying to explain also the need and show how we can bridge a tremendous need in public health in many, many countries, actually, with a technology that is compelling in the sense that not just the science, but also the cost. Paul and I spent almost 10 years doing a lot of DNA, cell-free DNA sequencing, circulating tumor cells. We did, you know, a tremendous amount of work, and then we found that it had difficulties translating into medical practice. It was difficult to bring that to the patient. So Hawkeye is very much also about getting this into the market, not for the obvious reasons of creating, you know, revenue and generating some returns for our shareholders, but also putting something into the hands of primary care physicians and patients, because disease avoidance is the name of the game in lung cancer. So I'm a little bit more commercial than Paul, obviously, and you could tell he was using slides that were not entirely his. So I'm sorry, Paul, but thanks for delivering the talk.
Great. And Haley from ARE, you have a question.
Sure. Yeah, I was just going to ask about whether you guys have a sense of what the sensitivity would be for residual disease monitoring and going after that patient segment.
Yeah, that's a segment that we're very interested in, and it's a subject that we discuss on an almost daily basis. We haven't yet done experiments with MRD, so I don't have a hard and firm answer for that. What I will say is in the data that I presented briefly here in the 351 patients, we had a quarter of those patients with cancer had stage 1 disease, and our accuracy in stage 1 is in the 90s. So you know, I don't know where the boundary is between recurrence and detection. But I think the performance we're seeing in stage 1 will predict that it will be useful in that setting.
Yeah, and then maybe just to kind of give me a better sense of competitive landscape, kind of how you see yourself stacked against some of the other, you know, liquid biopsy based approaches.
Right. Well, I mean, that's another big conversation. There are some very big players, some big elephants in the room that are very well funded. As Andre mentioned, we spent a number of years doing cell-free DNA sequencing, circulating tumor cell sequencing. And there are very interesting technologies that work very well in certain settings. It was not our experience that their performance was up to the detection at the earliest stage of disease where the survival was best. And so far, the data that's been presented is supporting that, you know, that remains a challenge. Sure. So the cell-free liquid biopsy will continue, I think, to develop. But having a low-cost, sensitive platform that is being analytically focused on the earlier stage of disease, I think, is it's a different capability. And I think we'll see more of these type of post-protein based technologies, as we see more and more people looking at the proteome and the metabolome. That's we're downstream of the DNA, and we're leveraging that sensitivity. Anything to add there, André?
Yeah, I would add the fact that we are working with proteases this time around. There are over 500 of them in the human body. We do have ambitions to throw different mixtures of proteases at panels. So to the extent that GRAIL, Thrive, and all the others are promising panels in the future for age 40, 50, 60, et cetera, sort of the moving beyond the classic liquid biopsy that we've been doing for 10 years, I think you'll see us as a player there as well. We have early data in beyond oncology as well, which we're very interested in doing, and we're going to be talking next year about a large 20,000 patient study that has not yet been published, but does look at these different signatures. So we like panels, we like liquid biopsy, but we also like things that can be performed for a few $100s on a routine basis for patient care. I don't think you'll see us in the 10 to 20,000 liquid biopsy lineup, so to speak.
Great.
Thanks.
Great. Just one last question before we kick it over to Steve and Andy to wrap things up. Question to both Paul and André. You mentioned about the Series A at the end of the year. How much are you looking for, and what kind of use of proceeds?
Yeah, sure. So most of the, I'll tell you what we raised so far was 7 million euros at the end of 2019. We did base the company in Ireland, and this was heavily influenced by what we're doing in China. And so, and the political climate, it's really nothing more than that. I just want to outline that it's not a tax play or a IP inversion or anything like that. Today, our need for capital is driven by a fairly ambitious clinical program of up to 20,000 people. So we're going to be looking for $15 to $20 million, possibly in tranches, but mostly to fund the clinical work and to fund a conversion to GMP. In fact, that's why I couldn't be, on the call in person. I have to come down and talk to some companies here. That has, a certain fixed cost. So that's most of where our budget comes from for the raise.
Excellent. Thank you very much. And great presentation, Paul. Greatly appreciate it. And to all the presenters, thank you very much for your time. And without further ado, I'm going to turn it over to Steve. I don't know if Andy was still online, but Steve, I'll turn it over to you to bring it in.
Oh, great. Thank you, everyone. Thank you, David, for a wonderful job moderating, really helping to translate all the pieces for us as well. It's great. Very amazing. And as Dave mentioned, we are recording this. I will distribute to those in an email follow-up. And of course, if you are interested in connecting with others, please do feel free to put your contact information in the chat, and I can distribute that to everyone else. If you did not want to put yours in there, that's okay too. But if other people do want to reach out or connect, I'm happy to make that connection as well. I will play the matchmaker, so please feel free. And with that, Andy, did you want to make any last remarks?
No, just to echo your comments and to thank the researchers and the presenters. And feel free to email us. This is a program we're actually doing every month. We're alternating between life sciences and kind of traditional tech. Our next month is on applied robotics. Probably not relevant to many people on this call, but if you're interested, let us know. And if you have topics that you think are of sufficient kind of breadth and depth, and you want us to calendar those in terms of future events, we're also open to suggestions. And be assured, once you're on our list, you're hard to get your name off of it. So we'll keep you engaged. But really, very impressed. And I know I was getting a bunch of direct messages for people who know the space better than I do, and they were very compelled by the science. So keep up the great work. We're counting on you to make the world a better place.
Great. Thanks, Andy. And for that, we can give you back a minute, right? We're a minute early. So there's something said for that. So thanks, everybody, and appreciate your time.
Thanks.
Thanks, everyone.
Thank you.